Good afternoon, my name is Quinn Remich, and I'll be presenting my group's findings for you. Now, our question was, how will the variance in salt concentration affect how well trypsin, a protease, breaks down as a casein, which is the substrate in this scenario. Now, a protein is a string of amino acids that have specific subgroups on them. These subgroups determine the shape of the protein, and then this final shape of the protein determines what the protein will perform. Some proteins are structural, some proteins are digestive. Trypsin is a digestive protein. It's an enzyme. An enzyme is a protein that jumpstarts reactions. This means that a reaction needs less energy than it normally would. And this is important because in our bodies, we need to be able to our body needs to perform chemical reactions without using a lot of energy every single time. So these enzymes help with that. Now, trypsin is specifically a protease. It breaks down proteins by performing hydrolysis, which means it takes a little water molecule and it puts it into a chemical bond, and then that chemical bond splits in two. One part gets a hydroxyl group, which is HO-, and one group gets a hydrogen ion, which is H+. Now, as a casein is a milk protein. It's just a dietary protein that gives our bodies the nutrients that it needs. It has something attached to it called sulfanilamide. Now this allows azacacein to absorb light at 440 nanometers. Because of this, we are able to test how well the trypsin has broken down the azacacein. Now we're going to be changing the salt concentration to see how that affects whether or not, or how well azacacein works. The 4 salt solutions we used are 0.75, 0.15, 0.3, and 0.6 molarity. Our hypothesis was that 0.3 molarity would have the highest absorbance rate, and this is because in our last experiment, that is what happened. 0.3 molarity had the highest absorbance of all the tests we did. It was, we do not know whether or not this was a significant difference, but it did have the highest absorbance rate. Now, the controlled variables. Each of these salt molarities are going to have things that do not change in any of the experiments. Its temperature, incubation period, pH, centrifuge time, speed, quantity of each buffer, and the quantity of enzyme and substrate. Now, we kept all of these the same in each experiment because we only want to know how the salt concentration will affect them. We were advised to use the temperature that other groups found was most effective. And 47 degrees was approximately what other groups found was the best and the most effective out of all the other temperatures they tested. When we were looking at pH, all the salt solutions that were given to us were all the same pH, so that was kind of controlled for us. Our methods, as you can see here, we started with 0.075 molarity. We first added it to 4 clean centrifuge tubes, so we're setting up 4 tests. We added 500 microliters of azacacein, preheated the centrifuge tubes for 2 minutes at 47 degrees Celsius. Now the reason we did this is because once we added the trypsin in, we wanted to start it incubation immediately, so that once it goes in, we're timing it for exactly when it starts digesting. And then we added the 50 microliters of trypsin to each centrifuge tube. And we incubated each of the tubes for 20 minutes at 47 degrees Celsius. And we want the time to be the same for each of the trials, because again, we're not testing time, we're testing salt solution. This is partly why we preheated them and why we timed very specifically, as much as we could, each of the trials. And we did the trials one at a time so that we could have as high of an accuracy as we could. We incubated for 20 minutes, and then we added 500 microliters of TCA. Now the reason we did that is because TCA is an acid. it will stop the reaction of the trypsin, and it will break down any of the rest of the trypsin and the casein, making it all fall to the bottom. This is also for when we centrifuge it. If we did not have the TCA, the casein and the trypsin would just stay in there and they wouldn't fall to the bottom. But because we have the TCA, they did. We centrifuge the tubes at 12,000 RPM and centrifuge for 2 minutes. We then transfer the supernatant to a clean cuvette. This is because we didn't want any of the pellet in what we are reading the absorbance for, because the trypsin, the casein, we're not reading the absorbance of that, we're just reading the absorbance of the supernatant that's left behind. This will show us how well the trypsin digested the casein, because the casein that's digested will not be taken out by the TCA in the centrifuging, it will stay in the solution. But anything that wasn't digested, all the trypsin and anything left of the casein will be left in the solution and the supernatant. We then read the absorbance on blanked the spectrophotometer using a cuvette with DH2O, distilled H2O, so we made sure it was completely clear. And then we read absorbance AU in the spectrophotometer at 440.1 nanometers. This is, as I explained earlier, because casein has something called sulfanilamide. This will absorb light at 440.1 nanometers. This will allow us to get a number of how much casein is left in the formula. We then repeated procedurally each of the steps for 0.15 molarity, 0.3 molarity, and 0.6 molarity. After this, we repeated all of the steps again for each of the salt concentrations, but this time we did not add any trypsin. We did negative controls as well. We did this because we need to make sure that the tests we're doing are specifically testing the effectiveness of trypsin. If we did the negative controls and found that all the results were the same, that means that trypsin is not being affected at all. It's not affecting the results at all. This helps us know